Recap. So genes, proteins, come into life. Are cells, is DNA as the information. The way the information flows is from DNA to RNA to proteins. Proteins are the machinery of life. They do everything that is done. And in terms of understanding sort of the complexity of life, or the way the machinery functions, there is the 1D, essentially this is an approximation, but mostly true. There's a 1D sequence that contains all the information from DNA to RNA to proteins about how the three-dimensional structure of a protein looks. The three-dimensional structure of a protein contains essentially all the information about how it functions. When I say essentially, so there's a little bit of the caveat that it depends on what is next to you, in what environment you are, what you can interact with. Going from the 4-letter alphabet of the genome to the 20-letter alphabet of the proteins, we need codons or triplets. So triplets of nucleotides, 3 nucleotides that together code for 1 amino acids. They're referred to as codons. There's large-scale sequencing. So one way to imagine that, the printer that we all know produces about 6 terabytes of data and sequences the human genome every day. So these machines are of that size, and they're much bigger machines that produce much more. The proteins are made up of amino acids. You can essentially imagine them to be pearl chains, where the pearls have different colors, 20 amino acids, 20 different colors. And these colors come with certain features, and we're going to talk about it a little bit more today. In people, every one of us has about 20,000 proteins in each cell. They are not all activated in every cell. So we have about 200 different cells, and they do different things, and they have a slightly different repertoire of proteins. But all of these cells together, that's 20,000. So our genome codes for roughly 20,000 proteins. And of these 20,000, now it's said 2 groups in the world have seen 16,000 in their lab, so have lab evidence. And from the error estimates, they can estimate that the number will be 20,000. But experimental evidence is available for, that depends a little bit on how you say this, but roughly at most for half. When you ask for how many of these 20,000 proteins do we really know every detail about their function, then that number is very tiny. Tiny means about 1,000, so it's still a large number of proteins, and this is because it's very, very difficult to determine in detail how a protein functions, and this is why protein prediction is so important. Today, what we know, when I say 85 million proteins, what I want to say, or what I mean by that, is for 85 million we know the chain, the sequence. We do not know what they do, we do not know the structure. They differ in their length from 30 residues to 35,000 residues. So they span an immense amount. And one way in which you can visualize this immense span is the different scales of protein structures that you see here. Another point that I alluded to that is very important in the way you imagine a cell. The cell is very dense. So this is an image of a cell from David Gotzel, where the green around here is the lipid, the membrane, the wall. If you imagine the cell to be a medieval town, there's a wall around it, that's in green here. And inside there's a high density. It's the density of a solid state. It's not a liquid. There's a lot going on. So picture it like a cog, like a watch with cogs and wheels. So today I'm going to talk a bit about amino acids, protein structure, bonds and energy terms, and domains, and a little bit about 3D comparisons. Next Thursday, again next Tuesday will be skipped. Next Thursday we will go into so-called alignments, so how to compare the strings, the 1D strings, the sequences. And when I talk about protein structures, one important issue here is about the way we imagine the reality and what the reality really is. We build images, and this talk or this idea comes from Marco Punta, shown here. That is Marco. Wait, there's a larger image of Marco here. What's that? What do you see?
Tree and houses, maybe.
Tree and houses, Georges Braque, houses at Lastac. So tree and houses, this is essentially what you see there, some stones. So it is up to you. And now many people here in the room most likely have seen something like that. Okay, maybe some have seen E. coli. Because I've been asking for E. coli so many times, or a virus. But ultimately, what he paints gets in you this idea that you see houses, trees, that you see landscape. Although if you really were to delineate what is what here, it's a difficult task, right? So there is not a one-to-one in what you see and what you internally see. So what you can see in a computer, what you could see in a computer here, and what your eyes see. What's the difference? The difference is sort of expectation. What the image triggers, and that is at the base of what I mean here by the difference between images and reality. So I already told you that is a cell. In fact, this Mycoplasma genitalium is one of the smallest cells you have. That's a cut, a slice through a eukaryotic cell here, a little bit larger cell with different compartments. What is that? Please louder.
Protein?
EM. EM, what is EM? So let me explain. I stand here like an idiot. Somehow I hear things whispering out of from my ears, something whispers out of somewhere. I know roughly where it is, but I have no idea who said it. What is EM? Whoever said it. Proteins. Is that clear for everyone? I hope that at the end of today's lecture you will see something like that and you will recognize it as a protein, at least on that level. Now what I'm going to show you is the same reality, the same protein, in the same view, in a slightly different way of showing it. This one. I flashed in the last lecture on Tuesday, some of you may have been in the room, I flashed already a protein and I talked about the rods that are helices. You immediately recognize helices here. You immediately recognize these rods. You immediately recognize, even looking at these rods, that there possibly may be a symmetry between blue and red, that there may be a similarity between the two. So suddenly you begin to see a lot of other things. There's a different way of showing this. This now comes with names. Alcohol dehydrogenase, ADH, is a particular protein, which is a so-called homodimer. Dimer means two of this, and homo means the same thing again. So essentially what you see is one unit here and another unit here. It's the same thing repeated. Essentially the same protein coming there twice. And it has to act as a dimer in order to be functionally active. So if these two things don't come together, two times the same protein, then this will not digest alcohol. And you will feel very bad after drinking the beer. What's that? And now I need a few more people. So in this particular case, this is no longer as simple as the houses and the trees. I like some associations. I like some ideas.
Flower?
Flower.
Fire?
Fire. There was something else here?
Cloud, maybe?
Cloud. Oh, you're talking about this one. You're walking too much in the area here. Actually, today is sunny. Anything else? Plant or flower, fire, cloud. Nothing else? Sorry, I'm hearing back. Paper? Paper. Oh yes, use paper mache pushed together. Yes. So somehow there's only one aspect here that I understand the paper, I understand the flower, and I believe you two have a similar idea. The clouds I see too. The thing that I was after here was more the fire, because what you actually see here. Read the title, and it's quite obviously that you see a soccer player. In fact, you do not see a soccer player, you see the dynamics or the dynamism, so the degree to which a soccer player is really moving around. And here is the real soccer player. Incidentally, that team played yesterday. But in some sense, so the other team, the defending team here, in some sense, if you look at this photograph, okay, okay, in this particular group here, only one person had fire. Typically, more people say something that has to do with energy or burst or some activity. And in some sense, I would argue that it's easier to see sort of dynamic activity in this one than in this one. This, in some sense, clear. This is a soccer player. There's no doubt about this. But the dynamics of this image, the real, or whatever you call real, this image here about the soccer player is frozen. And the way we look at it, we don't see how he's moving. If he leaned over a little bit more, maybe you began to feel that he falls. So you would not really see that he's completely full of energy and is doing something. And that is his trick to get around the other player. And this is what I'm sort of trying to say here. In proteins, it's the same thing. So these are, again, two different, slightly different versions of the same protein. It's not the one I showed you before, it's another one. And last time I showed these, what now is here sort of greenish or turquoise. Last time it was in yellow. Those are the fingers, beta strands. And there are these arrows here. The arrows point in a direction. That's the direction in which this chain runs. So it runs here, then there, then here, then there. That's the way it works. So it's, again, I cannot do that with my fingers. I would have to turn them around. Every other finger. But so this is one way of looking at it. This is another way of looking at it. And both are different realities of the same thing. Okay? Now, in the left one of looking at it, here in this one, when you look in more detail, I'm not entirely sure how much of that you see in the back, but you see that there's sort of, you may see that there's a blue here, and there's another blue here. So there are different lines. Actually, these different lines are different models or different structures of the same protein at slightly different time points. Or it's more complicated what really is. It's different models, different solutions to the same problem. And when you see that all the blue lines converge to the same point, then that's where it's more static. When there are different blue lines, in particular when they diverge as much as here, then essentially there's a lot of movement happening. So that's very dynamic. Those areas are very dynamic. And again, the original file that produced this plot were identical between these two. The original data is identical between these two. These are two different realities that you essentially can extract from the measurement. And the measurement is sort of an extraction of the reality. Okay? So we have a similar situation. We have an abstracted reality. We have an abstracted image. We create an image, and then we believe, looking at that image, whether it's this, it tells us something. And it does. We learn something from it. We have to be careful. What we learn is not reality. None of the two is reality. Not this one and not this one. They're both approximations. They both tell us something about reality. Okay, let's go into the constituents of proteins, amino acids. The simple structure of an amino acid, one way of simplifying it, is to say there's sort of a main part referred to as the backbone, and a sort of minor part, the side chain. Now the side chain, so major and minor, again in our head typically we would assume, and the way I plot it here, side chain is small, backbone is big. For many amino acids it's just the other way around. But this is the part that all 20 amino acids share. 19 of them also share that they have a side chain. One doesn't have a side chain. But this part here is really the backbone that is shared by all of them. Now, when we join these two, when we join two amino acids into peptides, so in the single one here, dipeptide, so two amino acids together, then typically the side chains you see are sort of on opposing sides. And here you form what is called a peptide bond. This peptide bond creates water. This peptide bond comes from sort of two principles. There's an attractive force and there's a repulsive force, which in physics you would describe by an energy function here, where the distance is here, the energy height is here. So there is an optimal point here somehow in this minimum. That's where most of these things would bond. Most of these two amino acids would bond in a way that their distance is exactly in this point, okay? You get closer, you have the electrostatic repulsion, you get further apart, you have an attractive potential. Now again, even this bond, one way of looking at this bond is this sort of space-filling model. So there's a probability how this molecule looks. The highest probability is where you have the big spheres, and then there is the connection in between. Depending on the motion, this changes slightly. So the spheres may be bigger. Then depending, again, on the way you draw that, it may look more like this and show the electron density map around it, which essentially is the part that is somehow could be influenced by other chemical activity. So again, we have 3 different images, or 4 different images, counting this one here, or 5 counting this one, and they all convene a different aspect of reality. All images. And then we keep going. So in this particular case now we have 4 amino acids joined together. Just going back here to assign what is what. This is here, this is that part of the blue, and that part is blue, is that one. And then again, when we join them, you see that the side chains tend to sort of alternate. This brings us to this issue of, I like to call it rationalizing biophysical features. So these 20 amino acids have features. And these features are essential for forming 3-dimensional structures, for the way these 3-dimensional structures come together and the way they look, all right? But our understanding of these biophysical features, again, is a very crude approximation. These are tendencies, more or less, not it is like that. These are not definitions. So let's begin with side chain properties. These are the 20 amino acids and their names. 2 of those 20 have positive charge. Okay? And even saying that one, there's another one that can be induced to have a positive charge in certain environments. The R and the K, the other one would be the H, the histidine, arginine and lysine. Then there are 2 that have negative charges, D and E. Then there are some that are polar, so they have a different charge distribution, so they have a dipole moment. Essentially, there are all kinds of other features. I didn't go into hydrophobicity here. Hydrophobicity means you want to stay away from water. Hydrophilicity, phil is the friend, phobe is being enemy of. So hydrophobicity means you want to stick together and be away from water, which in fact is what mostly explains the protein. So when you look at a protein, it is surrounded by liquid, by water. I told you that the cell is very dense and everything is as in a watch. Still, proteins are surrounded by water. It's just this idea that it's a little piece swimming in the ocean. You swimming in an ocean or even in a pool is wrong. But even if, what is another image that we could have? But if there even is a little bit of water, I'm looking for the image, I don't find it. I'll give you one later when I have time to think about this. But imagine a very, very, very dense pool. I don't know, a 10 by 5 meter pool with 50 people in it. Some of you may have been in pools like that. You touch a lot of people then, but you're still surrounded by water, okay? If you had a salty surface, that would melt, okay? And this is the same for the protein. So the protein is surrounded by water. Surrounded by water means that the outside has to be hydrophilic. It has to be residues that like water, while the inside has to be such that they like each other, they like to stick together. And that's essentially, some are hydrophilic, some are hydrophobic, and that's one way in which you could sort of predict how a protein structure looks by just putting the hydrophobic ones inside. One way to simulate. Now, I talked briefly about components. We'll be talking about it a little bit more. So let's go one step back. And what is called the gene model. So this here is essentially a very crude model for how a gene looks. It has here in front a region that is not translated. That means that will not lead to an amino acid or to a protein. There's an exon and an intron, so the red ones and the blue ones. Now the exons are essentially the only, introns are spliced out as it is known, meaning they are taken out. So the gene has the entire thing, and there are these blue regions that are there on the side of the gene, on the side of the RNA, but they are no longer there on the side of the protein. Only the red pieces here, the exons, only those survive into becoming the mature protein. So essentially the mature protein here, the gene product, the protein, is spliced together, as it is known, by these 3 red parts. How many parts this is depends really on the gene. These parts can be longer, can be shorter. Historically, there was the idea that these exons would relate to units in proteins. Again, I told you that proteins span from 35 to 30,000 residues, and that in order to do this, we need somehow a substructure, we need domains. And the first candidate for, when this was discovered here in the 60s, the first candidate was obviously these exons. They have nothing to do with domains, it turns out. But let's look at the evolutionary model, what we observe in evolution. For instance, as an example where we have in one organism protein A here, and in the same organism we have another protein, B, the green and the purple, and then there's another organism in which we have them both together as one protein. Okay? And typically that indicates that we actually have 2 different units. Because this unit, if I took it apart, it could sort of live alone. It is in another organism by itself, right? That is what I mean, live alone. What I simply mean is the protein adopts a unique 3-dimensional structure and has a particular function. This one, A, has in that organism, right? And the same is true for the purple one here. And that is the way we imagine these substructures, these domains. They are units that if I took them out and I put them into solvent, in principle it would adopt a unique 3-dimensional structure. And if they do, then typically I assume that in evolution we will see this often. See it again and again in different organisms. And maybe we see proteins built up in different ways of these things. Okay? And I'm going to show you some examples of that. In terms of 3D structure, an example is here, where this, again, you don't immediately see that. It takes much longer. Take my word for it. You have these arrows again, this time yellow, and I apologize that I sort of changed colors. So the 4 yellow arrows, the strands, in this particular case 3 helices, and that is a significant element that is repeated here, the much larger one. So it's a substructure. Okay? And in fact, when we look, and this is a slide from Christina Rengo here, so these are different proteins here, and these, essentially these symbols here, mark such regions or such domains. So one thing that you observe, there's not only 2 in them, so there are many, many more symbols here than 2 in each line. So there are more domains than 2. And you see that the symbols are repeated in different proteins, in different ways. So you often see this circle followed by a triangle, but then you see another circle in here, and then the way it continues is slightly different. It's all circles that it continues. Some have this kind of triangle, some don't, some have a different number of triangles, some do not end with the circles. So there's a variety of alternatives. You could imagine more combinations, and you actually see more combinations. Now, how could you, from the sequence, get at the identification of a domain? One idea is the following. I have 6 proteins here from, say, 6 different organisms. And what I mean is that the protein, when I compare these 6 by their sequence, then I can say that protein 1 starts here and ends here, and protein B, A here to here, B here to here. And in this region, where they are overlapping, where they are L-lined, they are similar in terms of their sequences. From the similarity in the sequence, I assume that they have a similar structure, and I assume that they have a similar function. Okay? I assume there's something here where they are similar. That's the way these, I will show several more lines, in particular next week when I get into the alignment methods, next week's. And you see then protein C, D, so you see different proteins, okay? And one thing that you observe is that at least 4 of them have this region here aligned, meaning they all have that in common. And it's sort of, for at least 3 of them, starts at roughly the same point. Maybe there's a little bit more, but not much. A little bit more in that direction, but not much. They also end somehow, at least several of them end in a similar point. And then you see something like that happening here again. So you would say this is one domain, this is one domain. Most likely this is one domain. This is at least another domain. Gentlemen? Any question? I'm happy to, let me repeat this. The lecture really is driven by your questions. And most of the time, what you believe is difficult for you only will be difficult for everybody else. This is new stuff. Almost every aspect that I'm going to cover in my lecture is for mostly 90% of the audience in this room entirely new. Please ask. Ask. The earlier the better. Okay, so, yeah?
Is the minimum length of a domain?
So a minimum length, let me be very radical, is 1. No, so this is nonsense, the answer. No, it's not really a minimum length of a domain. We know proteins that are 30 residues long, and we could call that a domain. Because they fold on their own, they function on their own, and they have all the features that I described I would want from a domain. Now, typically, people, so I show some work here from Christina Rengo, and typically people who build these domain databases, they look for things that are really repeated often in evolution. Meaning many organisms have the same thing. And typically those things are longer. So typically 50 and 60, around 50 to 60. There's a maximum too on the other end. So again, then things get a little bit apart. Do we call that 2 domains, 1 domain? But typically the longest domain, I believe that we somehow all agree on, is about 500 residues long. That's roughly the span of a domain, 60 to 500, which still is a factor of 10 almost, yes?
These residues that you're mentioning, they consist of, are they amino acids or something?
I'm sorry, I didn't make that clear. So the question was, the residues I'm talking about, are these amino acids? So, and I switched language here. I'm sorry for that. And I will keep doing this because my brain is too used to it, and the literature is too used to it. So an amino acid is essentially the idea of a single entity. It's an amino acid, okay? You join 2 of those, you no longer consider them amino acids, you actually begin to talk about residues. Yes, they are amino acids, but a bunch of amino acids strung together in a protein are called residues. Sorry for these words. I try to get the number of words as little as possible, but residue is important, yes?
Why is there a gap between domain 1 and domain 2? Why is that gap not considered?
So now we get into issues. So the question was, let me go a step back. Okay, the rationale here of calling this domain 1 is accepted. The rationale of calling this domain 2 is sort of accepted. But what about the region in between? And ultimately, the answer is, this is a region where the alignment is not so clear. So I put the second domain at a point that in some sense makes most sense. And I essentially say there's something in between where I don't know. So this means that domain 1 could actually be a little bit longer, or it means that domain 2 could be a little bit longer. Or it could actually differ between these 2 different proteins. Oh, let me go in this direction. So in 3D structure, one step more, these 2 things, I showed you that one before in a different orientation. So this is somehow contained in that one. Both of those are full-length domains. So they're not cut, they're not fragments. And this is an example. So it could be that actually some of these domains are a little bit longer, have some extra elements. Okay? So the thing in between could be protein specific, not sort of the generic domain. Again, this would imply by my way of thinking about it is you cut out between the yellow things, you put it into a tube, you get a 3-dimensional structure. Now the mistake in my definition then would be you could possibly add these 2 or 3 or 4 or 5 or 6 residues, a little bit here, this little bit here, put that into the tube and do the same thing. You also get a 3-dimensional structure that does something, okay? So then you would call this is a domain and that is a domain. Well, the common denominator is just the yellow part. Yeah? Thank you for asking questions. Yes?
How do you define the start of a protein and the end of a protein?
So the start of a protein is simply the first residue. Okay? In these images here, it's a little bit more tricky, but for a protein itself, it is made of N amino acids. So the first one is the start, the last one is the end.
If we turn it around, like the end.
So this is another word that we'll start with. Going back into, let me see how far back I have to go. the point where I showed you the formula of an amino acid. So this is called the N-terminal side, because here hangs over an H2N, and this is called the C-terminal here on this side because there's a C hanging over from the CO. So proteins are called N to C. N is the begin, C is the end, and ultimately on the side of the sequence we say when the codons are translated into proteins, the first one is the first, and that actually looks exactly like that. So because the peptide bond is always forming the same way here, and it's always oriented in that way. So the orientation, the moment you build the peptide bond, the orientation is clear. So now you could say, you could turn it around. No, you cannot. Okay? Because upon forming the peptide bond, it is clear what is historically first and what is last. In a protein structure, the story is different. So you can easily imagine that in a protein structure you should have the mirror image of the same thing, and the protein structure looks exactly the same. If it looks the same, it should function the same. This is true, and this is a remarkable detail. We will get back to that later. Yes, there was another one.
So these backbone parts, are they chemically identical to each other?
So these backbone parts again are chemically identical to each other. Yes.
Side chains are
the side chains, so the 20 amino acids have all chemically the identical backbone, and that's exactly shown here, 1, 2, 3, 4, identical, no difference, a different side chain. But the chemistry of the backbone is identical. And when we show protein structures, in one of the images that I showed you, that the first image where I asked you, what is that? And then the answer was, it is a protein with lots of things in there. Essentially what I showed is one circle for one backbone. So in that image I actually ignored the side chains. Most of the time you're ignoring the side chains. These images that I showed you there have 1,000 residues. So that's 1,000 spheres. Is actually high density of 1,000 spheres to show 1,000 spheres. And each amino acid has between, I actually can't remember how much the smallest is. The smallest has 10 atoms to, whatever, I have to look it up, 25 atoms or something like that. So they vary in size. So if you really would show every atom, it would get much less digestible. Ultimately, you only do that in binding sites of a particular DNA sites. Chemically identical are the backbones. All 20 amino acids are the same in terms of the backbone. Okay? Let's get back to the domains again. Thank you for your questions. This is really good for me and hopefully good for the lecture. Now, if I asked, how many domains does a protein have? The answer is like so many things in biology, not that simple to give. So one way of doing that is I look at the set of all the proteins for which we have experimental structures. At this point, there's over 100,000 of those, so I have a lot. But the ones for which I can do three-dimensional structures are sort of biased by having few domains, because then doing a structure, growing them into a crystal is much simpler. The bigger they get, the more difficult they are to experimentally work on. That's why if I look at the real three-dimensional structure, proteins of known structure, then I get an overrepresentation of single domain proteins. If I look, however, at proteins for which I do not have the structure, I'm not so sure about the domains. So you immediately begin to see where the complication lies.
The domains could differ based on the alignment of the proteins?
The domains can differ based on the alignment. So again, my operational definition is that the domain is defined as a region that is such that if I cut it out of the protein, I put it into a solvent, it adopts on its own a unique three-dimensional structure and function. That's definition point number one. Point number two is related to number one. If you have such a unit that will succeed in doing that, it's highly non-trivial. You cannot just string up, you build your own Pearl chain, most likely that will not fold. We will get back to that point. Most proteins that you can imagine do not exist. In fact, not most. The vast majority. It's highly non-trivial operation to create, to invent a protein, right? So if you come to that point where you have a domain that actually really does this, you take it, throw it into a solvent and it folds, then most likely it will be reused. So the second part of the definition then is you have a unit that you can see again. So this does not depend on the way the alignment is done. The precise definition where the domain ends and starts may differ, but the idea of a domain at least doesn't.
Can they overlap the domains?
So in this, the question was, can domains overlap? Yes, but actually, so we have experimentally seen examples where we have overlapping domains. And there's a longer story why, the way we explain that today. If I look at proteins, typically that's not what I see. Typically, so my definition, I can take many proteins and dissect them into their domains and put these domains independently into a solvent, and now I change these domains by adding I go a little bit longer, I see whether they overlap. And for proteins for which that has been tried experimentally, in fact, this has been done for about 100 proteins on many ways of overlapping. So for those, essentially almost never. So we have some for which it works, but it absolutely is an exception. We believe at this point. Yeah?
Is it possible to have a protein without a domain?
Is it possible to have a protein with what?
Without domains?
Is it a protein? So the question is it possible to have a protein without domains? Well, no, not really. So now we get into linguistics. When do you call a protein a protein? I would argue if you have something that folds on its own and adopts a domain. So by that definition, I'm sort of tricking, right? Whatever you call that, you call that a hack, my definition. But ultimately, so anything that is so short that it does not adopt a unique three-dimensional structure, I would call a peptide. So something that is of a fragment of a protein that is shorter, then it would have 0 domains, but then it would not be a protein. So by that definition, you cannot have a protein without domains.
You claim that having the 3D structure of the protein helps somehow to understand which parts of it are domains really, but how do they? I mean, if you're looking at a protein as a one single monolithic entity, how 3D representation explains better than 1D?
So I have to table that question. I'm gonna repeat the question, and then I'm gonna table it. So the question is how I say that from the three-dimensional structure I can learn something about domains, I can learn something about function. So the question now was how, looking at a three-dimensional structure, can I learn about domains? And I'm not going to answer that question because I went 3 slides to the left and I didn't see an example where I could show it. Maybe in the other direction I stumble over a slide where I can show it on the three-dimensional structure. Let me repeat this point that I mentioned before. So when we look at all the proteins, PDB, the protein data bank, is the proteins that contains all the proteins for which we know experimental three-dimensional structures in great detail, high-resolution structures. And you see that 60% of those here in green are single domain. Then 2 domains you see in, almost 30%, and everything else sort of drops off to very small amounts. In blue, you have the image that is an approximation saying, well, if you started from an entire normal protein, not from the subset of proteins for which we can determine the structures, but just take any 20,000, any subset of the 20,000 human proteins, then you would more likely see something like this. So most proteins, in fact, have more than 1 domain, and even most have more than 2. These sums look up in detail, and even further, this is still supposed to be an underestimate, so most proteins really have more domains. Let's get back to a different question. So when we talk about structures, I want to see a structure to compare function, then that would imply I want to look at a structure to sort of see domains. That would imply that when I compare 2 similar structures, they should have similar function. Oh, sorry.
About the
yes.
Wouldn't we need the data from the PDB database already to come up with these estimates about how the distribution of domains.
So the question, I'm repeating the question. I'm not sure I understand it, but I'm repeating it so far, and then I correct my understanding of the question. My understanding of the question is, when I know get a three-dimensional structure, again, PDB contains three-dimensional coordinates of atoms. That's simply a database of files that contain for every atom their three-dimensional position. From that you get the three-dimensional position of a residue, and from that you can create the images that I show you. And they're all available publicly, 100,000 or something. So the question I understand is, does PDB already provide the domains? Is that right?
Kind of. As I understood, we have in PDB, we have like guesses what domains could be.
Yes.
And the blue bars tell us like an estimate in all the proteins and
yes.
how many domains we would expect. But would we already need our guessed domains from PDB to come up with?
Yes. Oh, okay. The question is much more complex, and I'm actually not even planning to answer that in this lecture. I had, in fact, in last year in the same group, I still have answered that question, but this fell under the parts of things that I have been removing from the lecture. So, statement number one. PDB gives the three-dimensional coordinates. It does not give you an idea where the domains are. So the question is, can I learn from the three-dimensional structure the domains? I said yes, and I tried to motivate that, and there are groups optimized or who work their whole life on actually doing that. PDB itself doesn't do that. So from PDB does not come a domain organization. But then there are databases that use PDB and provide a domain. Now the question has been, how can I come to the blue here? So that would explain the green ones, right? But how can I take a human protein and guess the domains? Can I do that by taking sort of blueprints from the proteins of known structure? And that's exactly the way we did it. So we take a protein, and then we ask, in this protein, do we find anything that is already known structure? I cut this out. And then do we find anything else for which we have some evidence that it looks like a domain? I cut that out. And this way I sort of, this is why the method here is called chop. I chop the protein into fragments that I convince myself look like domains because of a variety of reasoning. This is not very simple. The idea is very simple, but the idea that you had is essentially exactly behind it. Okay, so let's begin with the matching shape. And I somehow, for me, this first slide here is one that I really like, because some of you may experience with children who are extremely intelligent, and then they suddenly come across a problem like this, and they try to put the green into exactly the hole as on this picture, and you may observe them and you sort of may begin to doubt their abilities. But this is something that when we are young, we really cannot do. And ultimately, this kind of problem is really not that easy to do on the level of protein comparisons. Well, let's do it simpler. Here is a mistake. There should have been a square, but anyway, so the match, so had it been a square, the match is very clear. Now we simply overlay, have corresponding points, very easy to do the match. Now if we had two objects that do not quite overlap, we still could define the matching points, and the points, in this particular case the green bits here, that don't match. Also relatively clear, relatively simple to do, and we can look at the differences. It's not that simple in detail, because there are alternatives. So you can turn around, there's some sort of symmetry operations, and they may matter or they may not matter. But ultimately it all leads to the point where you find the optimal superposition, and then you define or see the points, you label the points, and when you label the points, you can compute a difference. So for every single point in these two different objects, okay, you can do a root mean square difference. Simply, how far are they apart and what you call the best superposition, right? And this RMSD provides you the root mean square deviation provides you one measure how similar two different shapes are. So, incidentally, one problem of this is it is simply because you have a square in the number, you're going to be very sensitive to outliers. So if you have 100 atoms and just one pair lies very far apart, then this will matter a lot for the sum. Okay? This is just a feature of, is it right or wrong? This is a feature of this measure, and it has advantages and disadvantages at the same time. Now, again, that allows you to simply then define the region that you call matching. You say that as long as your RMSD remains below a certain threshold, you call it match. When you hit these outliers, or when you hit regions that make the sum no longer right, you no longer call it a match. So in terms of understanding, this is very simple. And this also means that now for doing an alignment, you first have to find the corresponding points, then you superpose, then you compile a difference. Trivial, very trivial. Here's the first issue where it gets complicated. So when I showed you the proteins, I immediately showed you examples where in fact the matching region was a fragment. The idea of domains is not that the whole protein has something in common. There are subfragments, there are regions, there are domains that in fact are such that I believe the three-dimensional structure will be similar. That means we actually have to somehow match these two. One way of doing it is, you sort of blow it up and see. But we cannot do that here. Proteins are defined the way they are defined. There is no scaling involved. So we really have to map residue to residue. We cannot sort of map on one 3 residues, on the other one a superstructure with 1 residue. It's bigger. So that brings us to the very simple issue that we will go through the following slides, which is global versus local. Global means essentially I take 2 proteins, I take 2 domains, I take 2 whatever, 2 units, and I compare them from one end to C. That's global. I make a comparison of the whole thing. And versus a local comparison, so when I compare these 2 objects and a global one, it's very difficult. So they don't have a very, very, very good global match, right? There are 2 global matches that I have shown here. Both of them are not very convincing. So you overlay very small subset of their units, of their domains. So the way these robots, or whatever you call these features, these professors and their dogs or whatever that is, overlay each element here is a domain. That's the way I look at it. Obviously, if you could dissect into these units, into these domains, then you could immediately see that after the dissection, you have a lot that you can overlap and that you can match, right? Immediately you see this. But this brings up a new problem. So in my drawing, it's clear where to cut, because I draw such that, this is the idea of the drawing. But ultimately the question is, what is the unit of the cut? I could say it's the domain. But then I already gave you this idea that the domain is not completely clearly defined. So it could be a little bit shorter. Maybe the shorter region is the one where everyone has the same. So we could still say I cut at the point where at least I could put it into solvent. But then maybe there's some overlap between domains. So this story gets more complicated. Even if in some cases there are overlap between domains, it sort of would ruin the generality of the definition. What is the unit for structural comparisons? Well, how could you decide on a valid unit? How could you decide when to cut or when to stop cutting? Again, we understand from these simple robot images that I have, global alignment is not the solution. We want to see whether 2 proteins that form a 3-dimensional structure are similar in shape, in 3-dimensional structure. We want to see whether 2 shapes are similar. We know the world proteins are organized in domains. We cannot do it in a global way. We cannot compare them, protein A to protein B. We have to somehow find the units, and units could be domains, okay? But in some cases the units will be shorter than domains. There will be things that I put into solvent and they will not adopt the unique 3-dimensional structure, but they will tell me something about a functional motive, for instance. Yeah?
You're talking about 3-dimensional structure, but inherently they are 1-dimensional, right? right? So this is just this
So-
expanded in 3 dimensions.
Is that at the end of the question?
Well, I have something to add. So if they are if you want to...
Let me answer the first part. So the beginning of the question is, I'm talking about three-dimensional structures. I'm talking about domains. I keep defining those things as three-dimensional objects, but in reality, they are one-dimensional strings. They're made up of amino acid or residues, and the reality of a protein is one-dimensional. No. The reality of a protein is three-dimensional. It only lives as a three-dimensional object. It has no, so in some sense, if you want, it's born as a one-dimensional object, but it never acts as a one-dimensional object. Any function that a protein has is a three-dimensional reality. So that first, is that a question to that? Oh no. So that first, so you just touched your hair. I'm sorry. So having answered that first part, how does the question continue?
Yes. So I'm asking actually information-wise. So exactly the same sequence of amino acids is going to always fall into exactly the same three-dimensional structure.
Yes.
That if this is the case, could we just use some sliding windows along the first dimension?
Okay, there's another method that will be skipped. So the question here was, I keep saying, you take a sequence, you put it in solvent, it forms the same three-dimensional structure. If that were true, why can we not just take the computer, take the sequence, and predict the domain? Yes, we could try that. And in fact, we have tried this for many, many, many years. We still, there's some success and some failure. And we will not talk over the course of this lecture about this, simply because it's another level of detail. It's not as simple as it may seem. It's something that is very urgent, it's something that we need, but we, as a field, have failed to do that. My group has has worked on that for over a decade, and has delivered things that sort of work, but not good enough to be of use for experimental, and that's why we stopped working on it. Okay, here's the question. A valid unit for three-dimensional comparisons. How do I stop cutting? How can I decide what a valid unit is? Any idea? What I'd like to do is that you try to form neighborhoods, and I understand that not everybody in this room is sitting in a neighborhood. And in this neighborhood, you sort of discuss for 1 minute your idea how to decide upon what a unit is for three-dimensional comparison.
Maybe try slicing the protein structure until it doesn't make any sense, until it doesn't have any function.
How do you decide? So you say slice the protein structure until it does not make any sense. What is the definition of sense?
Until it doesn't have any significant functional properties to hold.
So this is okay. We don't form groups. This is one answer towards what the group may bring. So the answer here is we cut the protein until we get to the point where what we get is no longer a functional group. It can no longer function as anything else. So one definition of that would be, I find something that is so short that I've never seen something that short to be specifically doing some function. And that doesn't exist. And so we have seen functional motifs that are very short. They are so-called nuclear localization signals. They are such that you put them onto a protein, and that protein is put into the nucleus. You can put them onto gold, and that gold piece is put into the nucleus by the machinery. These things can be 3 amino acids long. So the shortest one for which the experiment has been done with the gold was in fact 5, but some of these are shorter. So there can be up to maximally 3 units. That's statement number 1. So we cannot just have a size limit. Statement number 2, just a sec. Statement number 2 is we do not understand what these functional motifs really are. We cannot look at a sequence essentially exactly back to the question before. If we could look at a sequence and predict the three-dimensional structure from sequence alone, then we could do that, but we cannot. So this is another statement that I have to make. We currently know about 100,000 proteins experimentally. We cannot predict at this point for all the other proteins, human or in the world 85 million, the three-dimensional structure yet. And I will talk about why that is so difficult. But it is. Ultimately it's because the amount of possibilities that you have is too large. So you cannot completely sample the space, nowhere near it. Yes?
I don't know whether this is the case, but if there are some common patterns of how domains normally statistically start, maybe we find that pattern matching and cut at that point.
That is correct. So if we had patterns for the way domains start and end, then we could cut at those points and we could decide upon those points. But if we had those, we could predict domains. I already told you we cannot predict domains. So that is something that we had hoped, but by the way, here it's not necessarily, the answer is not necessarily domains. This could be that we look at structural motifs that are more related to function, that are more than or less than a domain. They would not fold on their own, but they would reveal certain functional motifs. And there exist some of those. And they are shorter than domains, we know that. But anyway, so ultimately, one way of saying is we cut and cut and cut until we find something that is somehow significant. It's the idea of we have a functional motif. It's the idea we can say that looks something like a domain begin, domain end. We see something that has some validity in terms of if I compare these two little objects here and I find some similarity, then this similarity tells me something about the biology. That's another way of doing it. It also means something we can look at statistical significance. So we can look at all the possible shops and simply ask which ones are statistically similar in some groups and not similar or different in other groups. This is a clearly operational program. Is not that simple to get through. Ultimately, we need machines that decide when to cut. There's something that has to do with scientifically significant, so something that is functional motifs are known, structural motifs are known, domains are known, and there's something about statistical. So I do this cutting for large data sets and I see something that looks statistically different or similar groupings. And I will do all of that. Then I will essentially do some cut, match, compare and measure by RMSD. That actually brings up the next issue of RMSD only looking at the coordinates and do a root mean square deviation, or is there more that we could do? What else could you imagine? So you have three-dimensional shapes that you want to compare, and don't forget these shapes have corresponding points, or they have residues, and these residues are like your coordinate system. Right? These are relevant. If you compare two three-dimensional objects, you have to remember that they ultimately overlay two residues. They are corresponding in two different proteins, right? So you will not have, these are 5 residues in one protein and 5 residues in another protein. The three-dimensional shape fit could not be such that that none of these fingers match. They must somehow match. Okay? So how can you do that? Not using RMSD, not using Cartesian space. What else could you do? Or not the trivial, you have essentially, think about it as a string in 3D, and you compare this, every single point of the string in 3D. What else could you do?
Model it by function.
You could model it by functional similarities, or by functional units. Is that what you said?
No, model, you said that it's like a shape or plane or something.
Yeah.
You could model it as a mathematical function.
As a mathematical function, so you no, we cannot, because these proteins cannot be written down as an equation. Because actually, you can string it up as an equation, but then you get to the same point, right? So the moment you build up so you string up a function that sort of has partially linear or partially definable things, and then you say from this to that is function 1, from this to that is function 2, and then essentially you do the same thing. Your protein of n residues would be strung up by n different functions, and then you compare essentially the endpoints, right, between two functions. So we're back to the same story. At least if I understand they're right. Yeah?
You could find some relationships within the protein.
So you could find some relationship within the protein. Yes, what could that be? So the simplest thing within a protein are distances. So essentially we move up the comparison from the level of essentially 1D. So it's no longer 1D because the string now is sort of curved in space, but in the comparison so far in the RMSD, we actually look at a 1D string in 3D space. And we could replace that by a comparison of distances, okay? This is one way of doing it. Yes, there was another idea?
Is it possible, for example, to, let's say, express mathematically the alignment of one protein with respect to another in terms of its rotation and displacement, and try to minimize this function? I mean, this.
Yes, but that is, so the question is, can we sort of describe mathematically the alignment between two proteins as a rotational and displacement? This is exactly what this is.
I mean, this could be.
But this is what we have here. But this is what we have. This is exactly what we do. This is a rotation and displacement. This is the way you find the optimal superposition. Yes, this is exactly what we do. The question is, what else? And looking at internal features, such as, for instance, a 2D distance matrix, is another thing. So I now replace the string, the coordinate points, by, in this matrix here, I put, for every single of these 8 residues, or one protein I have 8, another one I have 7, and I essentially replace the alignment of 1 to 7 to 1 to 8 in 3D-dimensional structure and doing an RMSD. I replace that by a matching of the 2D maps of the two proteins. The 2D maps that essentially, let me say that when you have a complete map of distances, you take an object with n points, and for this object with n points, for every single pair of points, you know how close they are in space. Then what you have is a 2D map of the 3-dimensional object, okay? Now this 2D map of the 3-dimensional object, one is 3D, one is 2D. Do you lose information? This is sort of yes or no. Who believes we lose information? Who believes we do not lose information? Sometimes it's difficult to see the minority. The majority in the room has no opinion, has no answer. The majority who did have an opinion said that there is a difference. Yes, there is a difference between 2D and 3D, but remarkably little. So when I have a complete distance map, the only thing that I cannot distinguish in terms of 3D structures is the image, or the object, and its mirror image. That's the only thing I lose. Which essentially is back to the question about the orientation. What is the beginning and what's the end? That's the only bit I lose. N and C. The label of N and C. What's the first and what is the last? Everything else is exactly identical and is completely contained. If you have a complete 2D map, it contains all information of a 3D image other than the mirror image of it, okay? So the directionality. That's the only thing that is missing. Which is actually, yeah?
Is the set of points limited?
In this particular case, the set of points is absolutely limited. Yes, we have n residues. N can be 35,000, is a big number, but it's still limited. Because the protein is limited, right? And the protein is not growing. In a different organism, there will be a different protein. Maybe in a disease state, the protein may be longer, but ultimately there's still a limitation.
But two proteins that you compare, are they necessarily the same size? Or
no, that's exactly the point. Two proteins that I compared, the question is, if I compared two proteins, are they the same size? No. That's the major point that I wanted to convene with this issue about global versus local. Proteins are not of the same size. Some are 30, some are 35,000. And a lot are in between these two sites. But even the ones, two proteins in the ballpark of 400 will not typically have the same length. And mostly, very often domains sit in two objects that have different lengths. So the same domain in these, I will not go back to the slide, but you may remember these lines that I had, the 6 proteins that I showed you, where I had domain 1 in yellow and domain 2 in yellow, they were all different lengths. And that is very typical. The domain is the same in a set of proteins, but the set of proteins differs very much in length. That's the tricky bit. That's why you have to look at local alignments. That means local alignments essentially means you have to dissect. You have to find units. And that is exactly the tricky bit. Anyway, so you could compare 2D, and I'm going to rush through a few slides. So here we have essentially an example of a 3D alignment. So we can align this one with that one, we can align that one with that one, we can compile the RMSD, we can see that A and B are similar, we can see that B and C are similar, but this, in fact, the triangular inequality is not given here. We cannot say anything about A and C. It's not a metric. So there's one alignment method that essentially uses the idea of so local environments, not quite distances, let's call it like that. So you look at a particular residue and its local environment distance and do a simple comparison of those. This is what is the background here. Then there's a method that essentially uses really completely 2D maps as shown here. So distance maps and the lines between two proteins to distance maps. So cuts this distance map into things that are statistically significantly similar in a large set of proteins and different in another set of proteins. So that's the way it sort of operates and cuts. The cut is then not done by expertise, what is function, what is domain, but simply what is statistically significant. Here's another idea that comes actually from Munich, Voronoi, which in fact replaces the residue by its Voronoi environment, which is somehow a similar idea as the distance environment. So you somehow describe something along your local environment that you believe is functionally important. So the way you're shaped, the way you're curved for a three-dimensional object is important, right? And this is what this one does. The major issue is always the one between local and global. Proteins do not differ in length, and then ultimately the question is, what is a better superposition? The one on the left side here, where some things get clear, or the one on the right-hand side, where other aspects get clear, and there clearly is no clear answer for that. There are many, many, many methods for 3D alignment of proteins. The point that I wanted to convey with these 3D alignment methods is there is a whole field in there. It's highly non-trivial. Ultimately, it goes back to your intuition about comparing three-dimensional objects. Ultimately, it goes back to the kinds of methods that people use for a 3D comparison of images in the computer, in the world, in the game world, is similar. It's just that the reality underneath is very different here. Again, cells are highly dense. Around, there's the wall. Inside, there's the so-called watch, mostly the protein machinery. Proteins come at different sizes, yes?
Have we found two proteins that have very similar but different functions?
Yes, so I believe in this one I don't have to repeat the question. That's the advantage of you sitting in the back, that everybody hears your question. Two proteins have similar, so we take a set that the same sequence leads to the same 3D structure. That's a statistical statement. Most of the time it's like that. There are exceptions. Exceptions are typically in a very different environment. This could slightly change. Then I said the same three-dimensional structure always does the same function. And the question here is, can I have the same 3D structure do two different things? And yes, that has happened. And now we, in fact, have two possible answers to this. In one aspect, you imagine that you so you have something of a flexible loop, and our antibodies do something like that. So they have something flexible and something rigid, and they can dock to different objects because they can slightly change this. Now I would, in this case of the antibody, I would define the main domain as the part here of my hand. I would say these two hands are similar, but the loops are slightly different. They will do slightly different things. So we would say, yes, they do different things. Okay, for the domain definition, they are similar. The whole thing is different, but then you could still argue, okay, but there is something different here. Yes. Now, can we have cases where the whole thing looks the same? Now, it's not quite that simple. The point is, I said that the loop here can change. But you could imagine that the loop changes upon binding situations. So depending on what is bound here, the loop actually may have slightly different features. So we unbound, we have the same. The moment this is bound, it's slightly different. Now it can do something else. Is that the kind of thing you mean? Well, that is one thing. But now let's take a case where we have the same two things. No bound, not bound, no alternation. The same two things, they do two different things. Yes, you can do that in two different cells, even in two different compartments. In two different compartments because you have different environments. In different environments, you can imagine that this loop is a little bit more open, a little bit more closed, depending on what the charge around it is. This is one example where we observe that. We also observe it, again, this is not quite this situation, but according to whatever you can bind to. So in one case, the protein meets DNA because it's a nuclear protein. In another case, it does not meet DNA and it binds lipids, because there are some features that lipids and DNA have in common. Now then you would argue, maybe, I'm sorry, I'll give a very, very long answer to your question. Is that now enough? My tendency is to answer, think about any exception and you will see that in biology. It's almost, well, no exception. So every story has an exception, so this statement also has an exception. But I don't know the exception to that one. Again, proteins come on very different scales. The shape of proteins are essential for the function. Here is an example. The way HIV docks, that is a slide from Natasha Wood from Cape Town. Here you see an image of the HIV envelope glycoprotein. So that's a protein that forms around the HIV virus inside of this bag or a sphere. You essentially have the virus DNA, RNA. And that docks onto the cell in order to put the genomic material of the virus into the cell. The docking is by recognizing this is a protein, a glycoprotein 120. This again, there's a glycoprotein that is GP120, glycoprotein 120, that recognizes particular structures on the surface of the cell that it wants to invade. CD4, CCR5. And these particular structures, they look in a particular way, and the envelope is always targeting the same. The way it's doing that, again, here's now the it's on a different resolution. That's the lipid here, the CCR5, the CD4. In fact, it forms a larger construct. In this larger construct, then it sort of injects the viral DNA. And this all can be stopped if we change this little loop. And that's a little bit similar to the story that I kept alluding to here. So this slightly floppy region that does not seem to have a well-defined three-dimensional structure because it can change according to this environment. The features of that loop are essential for this to happen. So you could imagine that we could simply engineer that, and this is exactly the kind of thing that people try to do. But this is an example where, in fact, even predictions of secondary structure, what is the loop, can help in the development of something that prevents AIDS from getting bad. Anyway, that's what Natasha is working on. We are back again to the three-dimensional modules, the domains, the so-called domains that are repeated throughout the proteins.